Cells need to make proteins. Enzymes made of proteins are used to help speed up biological processes. Other proteins support cell functions and are found embedded in membranes. When a cell needs to make proteins, it looks for ribosomes. Ribosomes are the protein builders or the protein synthesizers of the cell. They are like construction guys who connect one amino acid at a time and build long chains.
Ribosomes are special because they are found in both prokaryotes and eukaryotes. While a structure such as a nucleus is only found in eukaryotes, every cell needs ribosomes to manufacture proteins. Since there are no membrane-bound organelles in prokaryotes, the ribosomes float free in the cytosol.
Ribosomes are found in many places around a eukaryotic cell. You might find them floating in the cytosol. Those floating ribosomes make proteins that will be used inside of the cell. Other ribosomes are found on the endoplasmic reticulum. The endoplasmic reticulum has attached ribosomes and is called the rough ER. It looks like bumpy under a microscope. The attached ribosomes make proteins that will be used inside the cell and proteins made for export outside of the cell. There are also ribosomes attached to a nuclear envelope.
Every ribosome is divided into two pieces or subunits. In eukaryotes, scientists have identified the 60S, also known as the large, and the 40S, also known as the small, subunits. Even though ribosomes have slightly different structures in different species, their functional areas are all very similar.
For example, prokaryotes have ribosomes that are slightly smaller than eukaryotes. The 60S and the 40S model works fine for eukaryotic cells, while prokaryotic cells have ribosomes made of 50S and 30S subunits. It's a small difference, but one of many you will find in the two different types of cells. Scientists have used this difference in ribosome structure to develop drugs that can kill prokaryotic microorganisms which cause disease. There are even structural differences between ribosomes found in the mitochondria and free ribosomes.
So you might be wondering, when are ribosomes used in the process of protein synthesis? When the cell needs to make a protein, mRNA is created in the nucleus. The mRNA is then sent out of the nucleus to the ribosomes. When it is time to make the protein, the two subunits come together and combine with the mRNA. The subunits lock onto the mRNA and start the protein synthesis.
The process of making proteins is quite simple. First, you need an amino acid. Another nucleic acid that lives in the cell is transfer RNA. tRNA is bonded to the amino acids floating around the cell. With the mRNA offering instructions, the ribosome connects to a tRNA and pulls off one amino acid. The tRNA is then released back into the cell and attaches to another amino acid. The ribosome builds a long amino acid or polypeptide chain that will eventually be part of a larger protein.